the video for Lab 4, Streak Plate. The purpose of this lab is to first observe individual colonies. And a colony is a mass of microbes that is observable to the naked eye. And it is a mass of microbes that has been derived from a single cell that has been allowed to undergo cell division. And again, it is observable to the naked eye. Also, you will determine the colony morphology. So these are the characteristics of the colony, the size, the color, the texture. And all of these colony characteristics are determined genetically. So it will also help with identification of unknowns. And the third purpose is to correctly prepare a streak plate. So the whole purpose of a streak plate is to deposit individual cells on the agar and to give those cells time to go through cell division. So where you originally had one cell, you'll allow it to divide and form two cells, then those two will form four, four will form 8, and so on until you get a mass of cells derived from an individual cell, and this mass of cells is visible to the naked eye, and that is a colony. For colony morphology, there are several different terms you should know to describe different types of morphology. Form is how the colonies are on the agar. So punctiform is very common, and that's where you see a lot of very small colonies. And also circular is very common, where the colonies are just round circles. And then you can have filamentous, irregular, rhizoid, and spindle. Another set of terms refers to the elevation. That is if you look at the colony from the side, look at the agar from the side, and see how it rises above the agar. So flat means it's very close to the agar, raised, it's way above the agar, convex, it's curved, pulvinate means it's very high in the middle, and umbinate means it has a high center. And the last thing with this chart, margin, and that's looking at the outside outline of the colony. Entire means it's a nice even circle. Undulate means it's wavy. Filamentous is like hairs. Lobate is like lobes. Eros is serrated, and curled has different waves to it. Now some colonies are really distinctive, and these are just some examples of some really distinctive colonies. But these are not usual colonies that we will be working with. These are examples of colonies that you find from bacteria in the environment or from other unusual places. Most of the colonies you will see in this lab will have this sort of look. They tend to be white or off-white or yellow. So that's mostly the color of the colonies, and color is very distinctive too. Also, the colonies we are working with, the bacteria we're working with, are usually fairly small. They are punctiform or circular, but every difference you see in morphology indicates a different species. So of course, color, some bacteria can produce a pigment while others don't, and that's genetically determined. So that would indicate those are two different species of bacteria. Also size. You can have larger colonies and smaller colonies, and the size of the colony also indicates a different species, probably because the size of the cells are different. If you have tiny cells that are replicating and form a colony of 1 million, that colony will be smaller than a very large cell that has 1 million cells in a colony. So size and color also indicate different species. So any difference you see in size or color indicates a different type of bacteria in that colony. For performing the streak plate, you will need a Bunsen burner, a loop, the bacteria, the inoculum, and an agar plate. And to do this, the first thing you will do is sterilize your loop like you always do. Once the loop is sterilized, you will take a sample of bacteria, and remember, when you take the lid off the tube of bacteria, you flame the mouth of the tube. You take your sample of bacteria and then you streak it on the plate. So that is your first streak. After you have made your first streak, you flame your loop again. because what you're trying to do is dilute out the bacteria, so you don't want to add more bacteria. Once the loop is cool, then you do your second streak. After you do your second streak, you will sterilize your loop again and do your third streak. And this is what the streak plate should look like after it has incubated 48 hours. So your first streak is here, second streak is here, third streak is here. And as you can see, the streaks become less and less crowded with bacteria until the third streak, when you should get individual colonies. I want to go over the method in a little bit more detail on how to do the actual streaks on the plate. So of course you need your Bunsen burner, your loop, and your bacteria, and then here is your plate. And one of the first things you might want to do is to draw a T on the plate, because this can help guide you with your streaks, because you don't want to cross over into previous streaks. All right, when streaking with the loop, of course you take the loop, sterilize the loop in the Bunsen burner, and once it is cool, you take a sample of your bacteria. And again, just touch it to the bacteria. You don't need a lot, you're trying to dilute out the bacteria. So for the first streak, you'll start above the T and just take the loop and streak it back and forth across the agar. And remember, try not to dig into the agar, because you want the bacteria to be growing on the top of the agar, because you want to see colony morphology. Once you have made your first streak, then flame the loop again, because you want to kill off all the bacteria that are remaining on that loop. So for your second streak will be down here. And in order to spread out the bacteria, you're going to take your loop and start by picking up bacteria from that first streak. So you're picking up bacteria from the first streak, and then maybe cross over one more time and streak, spreading out the bacteria in your second streak. Then again, flame the loop to kill off the excess bacteria on the loop, and then you are going to make your third streak. And for this, pick up the bacteria from the third streak and make your second streak. And of course, the point of this is to separate out the bacteria. So if in your original streak you have lots and lots of bacteria all crowded together, hopefully in the second streak you will be spreading them out. And in the third streak you will be totally spreading them out, so you'll have individual cells separated from each other. Because after 48 hours, when these bacteria in the first streak grow, it will be so crowded that the growth will be like a solid growth, a solid streak of growth, so you can't see individual colonies. In your second streak, they're spread out a little bit more, so you may see colonies, but they're too close together to each other to get really individual morphology. But what you should see definitely by the third streak are isolated individual colonies. So that would be a successful streak plate. After your plates have incubated for 48 hours, this is what you should see. So this is an example of a good streak plate. And here this is actually 4 quadrants. So this was the first quadrant, second quadrant, third quadrant, fourth quadrant. And as you can see, it's getting less and less growth in each quadrant. And this is successful because you have nice isolated colonies, so you can view their morphology. This plate has good isolation of colonies, so it's 3 streaks, 1, 2, and 3 over here. And you do get isolated colonies, but this is not a good streak plate because there is also contamination. So you can see evidence of colonies with a different morphology, different size, different color, different consistency or texture, and that means that you have 2 types of bacteria on this plate. So for a streak plate, you want it to be done aseptically with no contamination. So this is not a good streak plate because it's been contaminated. Some more results. This is another plate, and this is not a good plate because you do not have any individual colonies. So these colonies are still too close together, so you can't see their individual morphology. And probably the reason for this is that the person, when they streak this, they went into the previous streak too many times. So they were picking up too much bacteria each time, and so they were unable to dilute out the cells far enough to get individual colonies. And another example of a streak plate is this. And this is definitely not a good streak plate because actually you don't see any dilution of the bacteria. So here's probably the first streak, here is the second streak, here is the third streak, and the growth is the same at the end as it was at the beginning. So what that probably means is that the individual went back and got more cells in between each streak. So obviously in this you have no individual colonies. This is not a successful streak plate.